Okay, so welcome to this next video on the Human Genome Project, in which we are discussing Sanger sequencing. Okay, so basically we have taken one of these bacteria from our genome library, and we are now trying to sequence the fragment of the human genome that it has within its plasmids. Okay, so we take these plasmids, we heat them up to 90 degrees to break the hydrogen bonds between the two complementary strands. We then take one of these strands and design a primer to it. Okay, so that primer then anneals, and I should say we cool it down after this. We cool it down to around 55 degrees Celsius, or below maybe, in order to allow the primers to anneal, basically. They won't anneal at 90 degrees Celsius, because obviously the hydrogen bonds broke at 90 degrees Celsius, so how will you get hydrogen bonds forming between the primer and the strand? You won't, basically, so you have to cool it down. Okay, so the primers bind to these now single-stranded plasmids, and what's going to then happen is you're going to put in a DNA polymerase enzyme, which is going to be allowed to synthesize a complementary strand to our green fragment here. Okay, now you give the DNA polymerase loads of different nucleotides here, and you also put into the medium terminator nucleotides, so terminator adenines, terminator thymines, terminator cytosines, and terminator guanines. Okay, so what's going to gradually happen is that because you've got so many plasmids that the bacteria has produced you, in each one of them it's going to be the DNA replication is going to be terminated at a different point, i.e. DNA polymerase will add in terminator nucleotides at different stages of sequencing the complementary strand. So you will get loads of different sized fragments, basically. In addition, what you do is you label the terminator nucleotides, each with a different fluorescent label, basically. So we've said we'll label the terminator adenines orange, the terminator thymines pink, the terminator cytosines green, and the terminator guanines blue. Okay, right. So now what we're going to do is we're going to again heat it up to a high temperature. So we'll heat it up to 90 degrees Celsius. Heat up to 90 degrees Celsius, and what that will cause is it will cause these strands basically to cleave away from the original strand of the plasmid. So you cleave the synthesized strand on the end of the primer away from the original strand of the plasmid. Okay, now if we think about all the different strands we have in here, what we will have is one where the first ever nucleotide you put in was a terminator nucleotide. So that's this strand here. So you have the primer and then just one nucleotide added onto it. Then you'll have the primer and then two nucleotides, the primer and three nucleotides, the primer and four nucleotides, and it will continue. You'll have the primer and n number of nucleotides. Okay, now what you do is you basically split these fragments up by size. So what you're going to do is you're going to get an agarose gel electrophoresis setup. So let's say this is our agarose gel electrophoresis. Okay, so you get a plate with agarose gel on it. You dig a little well inside of the agarose gel. So let's say this is a well inside of the agarose gel. And you put your medium containing all these different fragments in here. So this will contain the fragment where you were terminated after a single nucleotide, the fragment where you were terminated after two nucleotides, the fragment where you were terminated after 3, 4, 5, 6, etc. You put it all into this single well. What you then do is you put in two electrodes into this agarose gel plate. So this is agarose gel. I'll just put that up here. Agarose gel. Okay, you put two electrodes into this agarose gel. So one electrode over here, one electrode over here. Now, the electrode over here is going to be the positive electrode, the so-called anode, and the electrode over here is going to be the negative electrode, which has a fancy name again, it's called the cathode. Okay, so you then attach a battery between them to actually give them this electrical potential difference. So what the battery will do basically is it will produce an electrical potential difference between these two plates of the battery. So this small plate represents the negative electrode of the battery, and this large plate, tall plate here, is the positive electrode of the battery. So the electrical potential of this small plate will be some value, and the electrical potential of this large plate will be some value greater than that small plate. So if we look at E plus, if we say E plus represents the electrical potential of this large plate, and E minus represents the electrical potential of this small plate, then the battery will ensure that the difference between them, i.e. E plus minus E minus, is a certain value basically, which is the voltage of that battery. So it creates a certain electrical potential difference between these two plates. What's then going to happen is, because we have connected these two plates to these electrodes, this electrode over here, this cathode, is going to get the same electrical potential as this short plate over here. So here, the electrical potential of this metal electrode is going to be the same as the electrical potential of this side of the battery. And the electrical potential of this anode is going to be the same as the electrical potential of this plate of the battery here. So now there is an electrical potential difference between these two electrodes. In fact, the same electrical potential difference as there is between the two sides of the battery. Okay, now what can happen is that current can move through the agarose gel. So what you will get is you will get some movement of electrons from the cathode to the anode through the agarose gel. Okay, so let me denote this in blue. But more importantly for us, what you will also get is you'll get the movement of these DNA fragments that we've put in that well, because DNA has a negative charge. Remember, we're talking about single strands of DNA. We're not talking about double strands anymore. But if we look at the structure of a single strand of DNA, okay, so here is our sugar phosphate backbone, here's the phosphate, here's the ribose, here's our organic base, and then it will continue on, like here's the phosphate next, and then the 5' carbon, which I forgot to draw there, never mind. Here's the ribose, here's the next organic base. Okay, so these phosphate groups have a negative charge, so this has a negative charge. So DNA strands overall have a negative charge, so they're going to be attracted to this plate with the higher electrical potential, and they will move basically in this direction. Now, basically, we have put in strands of all different lengths, remember, into this well. Okay, so let me just redraw that over here. So we have our primer here, like so, and then we had the strand where you terminated the replication on the first nucleotide, this TT. Then we had our primer again. So we had, that was one of the strands we would have had in here. Another strand was the primer, followed by, you put in the normal nucleotide first, and then on the second nucleotide you then put in a terminator thymine. And then the next one, which was where you put in two normal nucleotides, and then the DNA polymerase just happened to choose a terminator nucleotide rather than a normal nucleotide, and then that terminated the DNA, and that was an adenine in the third position. Now, basically, longer DNA fragments are going to meet more resistance from the agarose gel. They're going to find it more difficult to move through the agarose gel. So, longer fragments move less in a certain amount of time, and short fragments move more. So, if we set this process up, we put in our battery, we set it all up, we put in our DNA fragments into that well, and we leave it going for a certain amount of time, these short fragments will have moved further than the long fragments. So what you will get is you might get, let's say this is fragment 1. Fragment 1 might get over here. Let's say this is fragment 2. Fragment 2 might get here. let's say this is fragment 3. Fragment 3 might get here, etc. Oh, what am I doing? Can't resist. Fragment 3 might get here. So the point is that this is going to separate out the strands in order of size. So you'll get the strand which had only one nucleotide added onto it, and that was the terminator nucleotide going the furthest. After it, in second place, will be the strand which had the first nucleotide put on as a normal nucleotide, and then the second nucleotide put on as a terminator nucleotide, and then it will continue basically. So the order in which these strands appear on this agarose gel plate will be the order will basically be the position at which the terminator nucleotide was added. Now comes the important bit. We fluorescently labeled these terminator nucleotides. So what we can now do is we can excite this agarose plate. So we can shine UV onto our agarose plate, and what will start happening is all the fluorophores will start fluorescing back at us. Now, let's just recall what colour we labelled all of these in. We labelled the adenine in orange, the thymine in pink, the cytosine in green, and the guanine in blue. So let's just recite that. So the adenine terminator was in orange, the thymine terminator was in purple, the cytosine terminator was in blue, and the guanine terminator was in green. Oh no, slightly wrong. Guanine was in blue. Okay, so this one was blue. This one was green, this one was purple, and this one was orange. Right, now what we need to also look at is the sequence of organic bases, which I wrote down as this sequence for green. So it was AATGG, okay? So I want to keep the example running well, so I'll use this. So our sequence was AATGG. So our complementary sequence to that would have been T, T, A, C, C. So this first strand is here basically. This had a terminator nucleotide of thymine in. So when we shine UV on, that's going to shine back at us as pink basically, indicating what? Indicating that the first nucleotide in this sequence on green must have been an adenine, i.e. it must have been the complementary entry to the terminator nucleotide that we are seeing was put in as the first nucleotide in this strand basically. Okay, then in the second one, again we're going to see pink, because this strand of second smaller size is the one where the terminator nucleotide went in at the second position, and the terminator nucleotide in that case would have been a thymine, so we'll see a pink coming through there, which tells us that the second nucleotide in this green sequence must have been an adenine, i.e. the complementary organic base to the terminator nucleotide which was used there. Then, in position 3, we are going to see orange for adenine. So, what that then tells us is that the third largest strand, well, the third smallest strand, which was where you terminated replication after 3 nucleotides, was terminated by a terminator adenine, which indicates that the third organic base on our green strand must have been a thymine, and you can continue this process on. The next terminator nucleotide, so for the fourth one, the terminator nucleotide would have been a cytosine, so it would show up green, basically. So we'd see green in position 4, which would indicate that the terminator nucleotide, which terminated you after 4 nucleotides, was a cytosine, and therefore the nucleotide in position 4 must have been a guanine. Okay, and in this way, you can sequence the fragments of DNA. So, this is why the Human Genome Project took 13 years and cost £3 billion, because $3 billion rather. They did this for every single fragment, basically. and then they had to painfully analyze, well, I think they got a computer to do it, but they had to build the algorithms to get a computer to then look at all this data, so look at all the sequences for every fragment that they had, and then what it had to do was find overlaps and piece the whole thing back together to get the whole sequence of the whole chromosome. So that's why it took forever. Okay, and in the next video what we'll discuss is next generation sequencing and how we have improved that. Because really, what we spent $3 billion on and 13 years of research was not sequencing one genome. I mean, that's not that great an achievement. What we did was went into improving the technology by which we were doing this to try and make everything work quicker.